Welcome, everyone, to NewLimit's 2025 Progress Update. The mission of NewLimit is to radically extend human health span. And we're a longevity company. We're excited about this area of research called epigenetic reprogramming. It's a way to restore the function in our aged cells that they had when they were younger. And the origin of the company really goes back to 2022. A group of us were sitting around thinking about what were the big problems to go tackle for humanity. How do we accelerate civilizational progress? And there was already really good teams out there working on AI, brain-machine interfaces, unlimited energy with fusion, maybe even going to space or another planet someday. But there was one area we felt like there weren't enough good people working on, not enough funding. And that was this idea of longevity. How do we take control of our own biology, our own evolution, to enhance the human experience and live more healthy years? And so we went out to see if this was actually possible. And we started by reaching out to the top biotech CEOs and scientists in our network and hosting these dinners. And we went around the table and we'd ask everyone, what is on the frontier in biology that you think has the most potential that is not getting enough research and funding? And several people mentioned this idea of epigenetic reprogramming to us. We went down the rabbit hole, we read every paper we could find on it, we met every person we could find on it, and I started to feel a little bit about epigenetic reprogramming like I did about Bitcoin in 2010. I just couldn't stop thinking about it. And when that happens, you got to start a company and try to make it happen in the world. So how are we doing as a species in terms of extending our lifespan? Well, this is the scorecard for all medicine and healthcare over the last 200 years. You can see that from 1850 to 1950, we learned how to wash our hands with soap. We invented antibiotics and vaccines. We made incredible progress. And since the 1950s, the rate of progress slowed a bit, but it's still going up. That's all modern medicine. And if you look at the very top right of that graph, you can see that actually in the last 10 or 15 years, it's kind of flatlined. We've stopped making as much progress, and it's even reverted a little bit there at the end. So the good news is, the last couple hundred years, we doubled our life expectancy, 40 to 80 years. How are we going to add the next 40 years to human life expectancy? Well, we think this is going to require a new paradigm, a new way to look at disease and medicine. And this graph was very inspiring to us. It shows that the biggest killers, the things that really take us out later in life, cancer, cardiovascular disease, diabetes, they're all correlated with age. That might sound like kind of an obvious statement, but it's actually quite profound if you think about it. When we're younger, our bodies can have damage, but we're resilient to it. We have the ability to recover from these damages, and actually most young people don't get taken out by cancers, cardiovascular disease, or diabetes. But as we get older, our cells lose their ability to respond to this damage or recover from it. And that's known as aging. Our cells lose function as we age. And so we had this kind of contrarian thought. What if aging was actually a root underlying cause of a lot of these diseases? And modern medicine had to shift from treating those symptoms to maybe figuring out the root cause of aging. So that's what we went out and decided to do. And this is an enormous opportunity. If there was a button that you could press that would give you the mental acuity, the energy, the metabolism, the freedom from aches and pains that we had when we were in our 20s and 30s, I'm pretty sure almost everybody on the planet would press that button. And so the total addressable market is enormous. The question is, can we actually go and do it? And that's what we're going to talk about today. By the way, I should say we're very inspired by the GLP-1s out there. These are Ozempic and some of these drugs. They've added over $1 trillion of market cap to Eli Lilly and Novo. And what's interesting about those drugs is that healthy people are taking them. It's not just people who have some disease and they get a therapy. That's the kind of the old paradigm of medicine. We're moving to this world where healthy people are going to start taking these in a preventative way or an enhancement way. And these are going to lead to more trillion-dollar drugs. So in a minute here, I'm going to invite up some members of the leadership team. And we're going to talk about some of the key milestones that we've hit just in this past year at NewLimit by working really hard. And then we're going to talk about some things we're going to do in the next year. So under the key milestones here, there's been a breakthrough. And number one here on this list, we've actually successfully demonstrated for the first time reprogramming older human cells to restore function that they had when they were younger. We did it in this case in hepatocytes, liver cells. We're going to talk more about that in a minute. Number two, our discovery engine has really started to hit its stride. It's scaling. And the hits just keep on coming. We have over 100 age reprogramming hits so far. We keep getting better and better hits that we now need to go run through functional assays. This has allowed us to build an enormous proprietary data set. It's about 500x, as far as we can tell, the rest of the known public field out there. That's allowed us to train the best AI models in the field to recommend the next set of experiments to go run in pooled screens. So we're going to hear more about this in a minute, and then at the end, we'll have Cathy, our head of ops, come up and talk about our objectives for the next year. How are we going to actually turn these into drugs? So now, let me turn it over to Blake, one of the other co-founders, and he's going to talk about, give you a primer on epigenetics and the theory behind why we think this is all possible.
Hi, I'm Blake Byers. I'm one of the co-founders here at NewLimit. It's a real gift to work on a project where you think success actually matters on a multi-generational scale. The thing is, working on ambitious companies that are failing is actually no fun. You want the right mix of ambition with reasonable chance of being successful. And when people hear you're working on a longevity company, they kind of like, oh, wow, that's amazing. And then they just stop paying attention a little bit because they're like, this isn't just never going to happen. So I'm just going to go about my day and not really pay attention to everything else you say. And I think this is like a relatively understandable perspective that people have because they think you're basically trying to break a law of physics. Aging is like entropy. It can only go one direction. There's no way to go back. So why even try? And although this is understandable, we know it's wrong. And the reason we know it's wrong is that you can already reverse major aspects of aging through epigenetic reprogramming alone. So you can take a skin cell from an old mouse, and you can, through epigenetic reprogramming, turn that into a newborn mouse with an entire life ahead of it. In fact, one research group did this experiment over 20 years, where they took a skin cell from one mouse, turned that into a newborn mouse, waited for that mouse to be old, took one of its cells, reprogrammed it into a newborn mouse, for 13 generations. And if aging was really some irreversible process, then that 13th generation of mouse would be a really messed up mouse. But it wasn't. It lived a normal lifespan. And in a way, this is kind of what humans have been doing for 100,000 years. We're going to have a 100,000-year-old cell line, and age resets every time we have a kid. We count age as on an individual basis, not a cell basis. If you're 20 years old and you have a kid, the kid starts at 0. If you have your next kid at 40, that kid also starts at 0. It's not like they got the next 20 years of your life deducted from their lifespan. So it is a kind of age reprogramming almost built into our biology. And that might just be a cute observation. I mean, there's tens of thousands of human genes and billions of base pairs. That sounds like a really complicated process. And then a researcher came along, Shinya Yamanaka ended up winning the Nobel Prize for this work, where you could replicate that whole effect with just 4 proteins. So these are specialized proteins called transcription factors that help turn genes on and off. And by adding these proteins for a number of days to any adult cell, you can turn it into a young embryonic stem cell. That cell can then give rise to young versions of that parent cell. And then other researchers followed on, like Marius Wernig at Stanford, and showed that you can turn one adult cell into other adult cells directly. So you could go from skin cell to neuron or liver cell directly to neuron. I want to pause here and just dwell on how dramatic that is. You go from a liver cell where your job is to process alcohol and caffeine to a neuron with axons and firing action potentials and releasing neurotransmitters. But there's one mapping here that's missing, and it's actually the most valuable mapping, and that's make an old cell into a young cell while maintaining its cell type or identity. So you can take that old cell and just directly turn it into, take an old liver cell, directly turn it into a young liver cell. In a way, this seems like it should be easier. We know how to take a skin cell, turn it into a neuron, ask it to do a totally different job, and it's capable of doing it. In this case, it's like, hey, take this old liver cell and just make it a little younger. Have it regain some of the function it lost as it aged. And despite it, as grand as a mission statement of NewLimit is, making humans live significantly longer, it almost sounds like it's unachievable. At the end of the day, it's a very narrow scientific initial first step. And that's refine these sets of proteins, these transcription factors that can restore youthful function in cells. Now, like anything in science, it sounds very simple. It's like, hey, let's just go find these. But the search space is massive. So if you just try to naively search it, it's basically practically impossible to do in a wet lab. It would require more sequencing than the world has cumulatively ever done. So you just can't go in and randomly brute force your way to success, or you'd be extremely unlikely to be successful. And so when we started NewLimit, we're like, well, what would be a smarter way of doing this? If we can't test everything in the lab, maybe we can come up with an engine that can screen at higher throughput through different steps and then progress down a funnel. And so we started the company, we put up basically this exact chart. And over the last 2 years, we've basically done all the steps, and now we're on the final step. And so I'm just going to walk through these steps with you, and then I'll hand it over to Jacob afterwards, who's going to be walking through the results that we've generated from this discovery engine. So the first step is AI models that we can use to assess cell age and also assess what factors are most likely to reprogram cells. We start with AI models because we can basically screen every possible combination with an AI model. We can then use those models to recommend what to test next. And that progresses to our look-like screen. These are basically gene expression profiles that we use to distinguish cells from old and young people. In the NewLimit labs upstairs, we have tens of millions of cells from old human donors and young human donors. And if you look at the gene expression profile of these cells, it's very obvious which cells came from old people and from young people. And so what we want to find are transcription factors that can make those old cells look like the young cells, have the gene expression of young cells. For the hits that we find there, they then progress to an even more accurate screen, and that's our ACT-like screen. So these are functional assays, and these are cell type specific. So for hepatocytes, it would be, yeah, are you doing a good job of processing alcohol or caffeine or responding to injury? For T cells, it might be releasing cytokine profiles that look more like young cells. And for the factor sets that pass that test, they move on to basically the final boss at NewLimit, which is to treat or cure a disease that we think would be predictive of a disease we would go into people with for our first drug product. And so with that, I'm going to ask Jacob to come up and start talking about some of the milestones we've hit with outputs from this discovery engine we've built over the last 2 years.
Hey y'all, my name's Jacob. Thanks so much for coming today. I feel really lucky to be the one who gets to share some of the results that our hardworking team has generated over the past couple years. And part of the reason I feel lucky to share these with you is that we're actually further along than I ever thought we'd be at this point, candidly. The first time we shared some of our work publicly, it was really just a series of ideas, and now we're starting to see some of the first hints that this reprogramming biology might actually enable us to make medicines that address some of the root causes of aging. And this is something I've really been working toward my entire career. I first became fascinated with this biology when I read a textbook when I was young and realized there was a chapter in the beginning that was super detailed about how all of us went from cells to organisms. And near the end, where I expected to find the chapter on aging, there was nothing. And there were no books out there. And realized that this is one of the most important problems in the world any of us could be lucky enough to work on with potentially the highest impact. And so I've been fascinated with this biology ever since, and for most of my career, I have not really seen the sorts of effect sizes, the sorts of progress that this new biology has unlocked more recently. So excited then to jump in and actually show you some of that data that has helped me get better sleep at night over the past couple of years than I did prior. One of the first milestones that Brian mentioned early is that for what I believe to be the first time, we've been able to restore youthful function in old human hepatocytes. So not only do they look like younger versions of themselves, but they perform functions like young hepatocytes do as well. And before I show you some of those results, I want to show you the system we've built that allows us to even ask those questions to make those measurements. So this is all hard work from our scientists here, where they've developed a set of tools where we're able to take old and young human hepatocytes. We start in human cells because we always want our experiments to be as relevant as possible to actually translating into human patients. And we take those cells and place them into a humanized mouse system. So this mouse is genetically engineered so that over time, their livers actually become fully occupied by those human cells. And so now you've got this really amazing system where you can study human genomes inside of a functioning organism. Now we can take old and young cells and put them into this assay and ask what happens. And what you find, maybe predictably to those of you in the audience, is that old cells are about an order of magnitude worse at actually regenerating in this context, repopulating this tissue the way they're supposed to, than young cells are. Now we hope this relates to some of the functions that are actually obligate in the context of human disease as well. So now that we have this system where you can actually measure dramatic functional differences between young and old cells that are related to human disease, we can layer in reprogramming and ask, can we find combinations of transcription factors that make these cells first look younger, as Blake alluded to, but more importantly, act younger, to actually rescue that function that's lost? And we can collect that with genomics measurements our teams have developed, where we can actually measure every gene that's expressed in these cells in a combinatorial way, and we can ask whether any of the combinations of these transcription factors we added to the old cells is sufficient to make them regenerate just like their young counterparts. And I'm happy to say that today we've actually found examples of both of those things. We found combinations of transcription factors that, when we turn them on, actually make old cells look like younger versions of themselves based on the genes that they're using. Here, we've trained AI models to actually discriminate cells from young and old donors so they can look at genes and predict how old was the person this cell came from. And we found combinations that even weeks after we last turned them on make cells look significantly younger. And more importantly, some of those factors actually make those old hepatocytes act younger too. They don't just express the right genes, they actually regenerate the way that young cells would. Some of these factors actually have effects that are large enough that these old cells behave pretty close to cells from donors that are decades younger than the ones we started with. So these are really just the first steps in our discovery funnel, and I'm showing you here, for simplicity's sake, a single experiment where we tested these things. But the next step in that process, as Blake alluded to, is asking whether or not some of those TF sets we discovered, some of those payloads, can actually reverse features of aging and disease in preclinical models. And so here I'm showing you just one of those models that we've used, where we can take livers from young and old mice and perform a very simple injury. You actually just cut the liver in half. And what you find is that in young animals, they regenerate beautifully. All of these mice survive. In just a couple days, they're running around totally fine. The livers are normal sized. It's kind of amazing how well the tissue works. In old mice, as you might guess, it doesn't work nearly so well. Unfortunately, many of them die. None of them regenerate as effectively. Now, members of our team were so excited about the results of that screen I just showed you, they actually took one of the top payloads and formulated it into a prototype medicine based on lipid nanoparticles and mRNA. So this is a type of medicine you can hold in your hand. Patients have been getting these LNP RNA medicines now for about a decade. These are the types of molecules that, while very early, one could imagine one day evolving on and eventually translating into the clinic. And we found when we give these prototype medicines to these old animals, we're actually able to significantly improve their regenerative capacity. You can measure this using specific molecular features of regeneration, or what I love, really simple measures, like you can actually just measure how big their livers are at the end and find that they actually just make more tissue. I always love experiments where you can know the readout without having to do anything fancy. And it's one thing to show you those box plots, it's another to look at the data yourself. And so here I'm just showing you an example of how we measure how well those tissues are regenerating. On the left, you've got a slice of liver from an old animal that hasn't been treated. And in this particular image, brown cells are regenerating and doing their job, and blue cells are not. And what you'll notice in the old animal that's untreated is it's really hard to find any brown cells, no matter how hard you squint. Whereas after we've treated with our prototype medicine, they're all over the place. You don't need to quantify it to recognize that something interesting is happening here, and some amount of capacity has been restored. Now, regeneration isn't the only thing that hepatocytes do. It's a very complex cell type in a complex tissue. One of the other roles that Blake alluded to is these cells are critical for processing the metabolites in your body. They store energy, they process environmental insults like alcohol and high-fat diets and caffeine. And we wanted to ensure that some of these reprogramming agents we're developing are actually able to rescue multiple functions. So we've developed another preclinical model here that I'm sharing with you, where you can likewise give young and old mice alcohol. And what we find is that, maybe like the experiment some of us may have run in our own lives, as these livers age, they're less able to tolerate that alcohol, and they experience dramatically more damage. Now, we can take some of those old animals and treat some of them with another one of our prototype reprogramming medicines, here just called M3, and we find that we're able to dramatically reduce the amount of damage they experience from that sort of alcohol insult. And so between this prototype medicine and the previous one I showed you, I want to emphasize that as far as we know, these are some of the latest stage results in terms of reprogramming that have really emerged in the field. It's one of the first times that we've been able to actually formulate a reprogramming medicine focused on trying to reverse cell age in a format that you could imagine actually translating down to the clinic. So I've zoomed in really far on just a couple molecules to hopefully convince you that there's some real function emerging here. But those molecules came from a much broader space that we've been searching through. As Brian alluded to, we've found now 100 or more hits. This is actually a little out of date. And when we say a hit, I want to talk you through what we mean, what leads us to actually get excited about a set of transcription factors. So what I'm showing you on the left is a plot where every set of transcription factors we've tested is a single dot. So the first thing I'll emphasize and brag about is there's a lot of dots. And I'm not the one who made them, so I can be proud of that. What we'll see is that on the x-axis, we can measure a cell's age based on gene expression. And on the y-axis, we can measure its cell type, where going up preserves the cell's original type, and going right means the cell looks younger. So up and to the right, like any good business metric, is really where we want to be. What you'll see is that some of the canonical reprogramming factors, like the Yamanaka factors, unfortunately, while they make you look younger, also push cells way down on the identity axis into that blue region, where these cells really no longer look like the right cell type to begin with. We can measure this using one of our AI models like we do on the left, or you can measure it another way. And what we find, for example, in that highlight on the right, is that whereas the Yamanaka factors make T cells look not very much like T cells anymore, many of the hits we've discovered don't seem to change the identity at all. And so we've now found hundreds of those hits that fall into that top right corner, where we think we can actually reprogram cell age while preserving cell type. And so even very early in the discovery process, we're trying to ensure that the payloads we're discovering are hopefully safe later on in the process. Now you might wonder whether or not we're actually able to restore function in more than just hepatocytes. And so happy to share with you as well some results from the team where it turns out we're able to actually see these sorts of effects across cell types, including in T cells. Now, T cells perform many, many functions in your body. They're very complicated. So I'm only going to talk about one today, and it's admittedly reductive, but I think still instructive as well. CD8 T cells need to clear cells in your body that carry foreign antigens. These are things like cells infected with viruses or cancer cells. And as you age, unfortunately, they don't seem to do it quite as well. So this might be at least one part of the story as to why infections become much more severe as you age. Now, we can also introduce reprogramming factors in that context, which was once another slide, but I will just jump through that particular set of dots because they don't show what we hoped, and jump straight to the real data. It's much more interesting to actually see the data itself. We can introduce reprogramming factors to these cells and then measure what happens when they try and control the growth of target cells that express a foreign antigen. Here in these movies, a red dot is a target cell that should die. So the more red in the image, the worse off those T cells are doing. What you'll see on the left is where we don't have any T cells. The image is quickly taken over by those red targets. In the center, where it's just old T cells alone, the red cells grow, unfortunately, quite well. But after we've reprogrammed them on the far right, we're actually able to see that these cells control the growth of those foreign cells much better. So we're able to rescue function not just in hepatocytes, but also in T cells. And these are just a few singular examples where we've seen those sorts of results. Now, along the way, those results emerged from a very large data set we've generated. To our knowledge, and this is really just based on what's in the public domain, there could be other data out there we don't know about, we've screened at least 500 times more in terms of the number of transcription factor sets for their effect on cell age than the rest of the field combined. And so we really think it's this scale that is to credit with the ability to make those discoveries that we've had. We're not necessarily better at figuring out which things to test as humans, but we are better at running much larger experiments. Now, we've really only been running these experiments at scale for about the past year. It took us a full year just to get this technology to work at all. The first time we shared what we were working on publicly, we were in the throes of trying to get this system off the ground. It was like an airplane with half a wing. And now, in the past year or so, starting at the beginning of 2024, every quarter we generate thousands of different TF sets that we test. And so really, it's about 100 times the scale of the field that came before us every few months. We're also getting much better at figuring out what to test, and you can see that in terms of the number of hits we discover every quarter over there on the left. We really credit that increase in the number of hits we're able to detect with the fact that some of the AI models we've been building, that I'll tell you about in a moment, are getting better at telling us what questions to even ask in the first place. Now, we've also built all of this technology so that it's general across cell types. Our ambition is not just to treat hepatocytes or to be a T-cell company, but to eventually be developing reprogramming medicines that address most of the pathologies that emerge with age. And your body is very complex. You've got hundreds of cell types. We want to meet that complexity as well. As just one example of that, all the hepatocyte data I showed you was really generated in just the last 6 months of the last year, and we hired our first employee on that team in about the middle of the year too. So really it was to credit the portability of that technology that we're able to bring a new cell type on board and get moving quickly. And we've tried to do all this while maintaining efficiency. It's one thing to generate a bunch of data. It's another thing to generate a bunch of data and do so on a budget. And so we've tried to really operate NewLimit based on some of the principles that tech companies operate by, which is really tracking rigorous input metrics, ensuring that you're always trying to drive down costs and not letting inefficiencies creep into your system. So over the past couple years, we've managed to drop the cost of testing the marginal TF set by more than twofold. And now all of that data doesn't just sit on our servers and lay latent, but it's actually allowed us to build what we think are some of the best AI models in the field. Now there are two types of model that we're really interested in. The first actually allows us to make some of the measurements we've already shared with you. It allows us to predict the age of a cell based on just its transcriptome. This allows us to ask questions like, if I reprogram this old cell, does it look younger? Seemingly simple on the surface, but surprisingly hard to answer. The way we've built these models is by actually building up a massive data corpus of hundreds of different donors and now profiling tens of millions of their cells and predicting the donor's age from those transcriptomes. And we do this in a fashion where we train on some number of people and we hold out some number of other people that the model's never seen before, and we measure performance on those new donors whose cells are totally unknown to the model at the time it was trained. We found the models we've built are more performant than the best baselines in the field, and that qualitatively, the predictions are shockingly accurate. When you look at cells from young donors, they get young scores, old donors, old scores, and you can show models things like cells from donors of a middle age they've never seen before, and they properly interpolate between the two extrema, as you might expect. Now, perhaps more importantly, we have another family of models, and I like to think of these models as the models that are trying to replace my intuitions, which I don't credit with all that much. So these models really take in a representation of transcription factors and a representation of old cells, what genes they're expressing at the starting place. And those models then try and predict what will happen to the age of that cell when we run a reprogramming experiment. We may have never seen those transcription factors in an experiment before, but if we can guess what would happen, we can prioritize what to do in the real world next. And those models are now actually quite performant. We can explain more than half of all the variation in our observations completely in silico. Now that's not perfect, but it also doesn't need to be. We're not trying to replace experiments with these models. We're trying to figure out what questions are even worth asking. Those models themselves are now, to our knowledge, the most performant in the field, better than the best baselines. And the credit we ascribe to that is really not necessarily to the fancy modeling tricks, though we have those too, but more so to the amount of data that we've generated and that our team has worked hard to build. We found that there's a scaling law in this particular problem, the same way we've seen in other domains in artificial intelligence, where the more data we have, the better the model performs. And so every time we run a new experiment, the performance of those models improves. And there's a cyclical feature here as well, where because the experiments we run are suggested by those models, if those models get better, each marginal data point is more likely to be interesting, and therefore the models get smarter as well. And so we feel confident that we'll be able to generate more value, more discoveries per dollar invested in the coming years than in the years that are behind us. Now, as I mentioned, we like to run NewLimit like a technology firm or a manufacturing firm where we rigorously track metrics, and we try and be really ruthless with ourselves to ensure that we're not only making discoveries, but doing so in an efficient way. I'm throwing up a few numbers on the left-hand side of this slide, not because I expect you to squint and read any of them, but merely to brag about the fact that we've managed to measure them at all. There are only two numbers I really want you to see. The first is that we've managed to screen about 50 times as many TF sets in 2024 as we did in 2023, and perhaps more importantly, I'm actually more proud of the fact we only scaled headcount by 30%. And so we're actually able to get super linear returns on these investments. Now, hopefully I've convinced you that there's some exciting discoveries afoot, that there's the bases for what could one day be medicines in patients. And to tell you a bit about how we'd navigate that journey on inventing medicines, I want to welcome my colleague Cathy O'Hare up to the stage.
Thank you, Jacob. Thank you, everyone, for coming. I'm going to talk about how we're going to get from where we are to developing medicines. So NewLimit's secret sauce lies not just in our science, technology, and talent, although we have plenty of all of those, but also in our focus on developing clinically tractable and commercially viable medicines. And this is the reason that I came to NewLimit. After finishing my PhD at UCSF and postdoctoral training, I transitioned into an industry career in translational medicine. Sorry, I'm getting a note to hold the mic closer. Translational medicine and drug development, where I've worked for the past decade. It is unique to find a company that is this ambitious in our ultimate impact, but also grounded in near-term value generation along the way. What gets me out of bed in the morning is the opportunity for impact, and this is a place where we can have impact now and a lot more impact in the future. We only work on cell types where we believe a reduction in cell age would make for a meaningful medicine. When you add them all up, we can impact human health span. But even before we get there, any individual program has an immense impact on human health. If we could reverse cell age in the immune system, we could improve immune resilience and control infectious disease. If we could reverse cell age in the liver, we could improve regenerative capacity and improve our metabolism. Brian has already talked about having your metabolism that you had in your 20s. That's why I'm here. If we could reverse endothelial cell age, we could improve the function of multiple organs, including the kidney, the heart, and maybe even the brain. What will our drug product be? For our initial drug product, and Jacob alluded to this already, we anticipate developing an mRNA medicine with a set of our reprogramming transcription factors packaged into lipid nanoparticles, or because it's easier to say, LNPs. There are commercial medicines available already that target LNP mRNAs to hepatocytes. The technology to targeted T cells is rapidly developing, rapidly advancing, and we expect future innovations to enable targeting to more and more cell types over the years. LNPs also represent a cost-effective delivery approach. LNP medicines are currently being manufactured for as little as $3 a dose. Lastly, the nature of epigenetic reprogramming means that even just transient expression of these mRNAs could yield durable effects. Brian mentioned the GLP-1s already. We are going to take a similar stepwise approach to what has been done with GLP-1s. Our ultimate ambition is to develop anti-aging drugs for a broad population, but we will initially develop each medicine in a narrower, established indication and then expand its use. GLP-1s were initially developed in a diabetic population. They were then expanded beyond diabetes to obesity and to liver disease. There were studies to demonstrate benefit broadly on cardiovascular outcomes. And now, as it has been mentioned already, there are patients or people, not even patients, people that don't even have a diagnosed disease that are taking GLP-1s. You can imagine a similar approach for hepatocyte reprogramming, where we start in something like alcohol-related liver disease, which is an advanced form of chronic liver disease, high unmet need, and patients have little to no therapeutic options. We could then expand into metabolic syndrome or multiple causes of chronic liver disease to the point where we're treating a large portion of the population. Similarly, for T-cell reprogramming, we could start in arthritis, we could move to infections, eventually we could have a drug that broadly improves immune resilience and protects people from a number of infectious agents. When we launch a program, we look for an initial indication where we can run trials in hundreds, not thousands of patients, and in months rather than years or decades, with an opportunity to then expand into a much larger patient population. So how are we going to do this? What's next for NewLimit? Jacob took you through the progress we've already made, and now I want to give you a sneak peek at what's coming over the next few years. In the coming years, we will have the first anti-aging drug ready for clinical trials. Not just the first anti-aging drug for NewLimit, but we believe the first drug built from the beginning to be an anti-aging drug. Not to diminish any of the work, the amazing work that Jacob took you through, the thousands of transcription factors, the millions of cells that have been screened, but they are just the beginning. All of that work fits in this first box of the drug discovery funnel. In order to get to our goal of making a medicine, there are still multiple steps that we will need to complete. So far, we've restored youthful function in old hepatocytes with our first prototype drug, and we're beginning to show that we can restore multiple functions in old hepatocytes with this prototype drug. Next, we will need to optimize the prototype drug or drugs that we want to take to the clinic. And this means figuring out exactly what the mRNA sequence is that we're going to want to use. Which LNP are we going to use? What size are they? Where are they targeted? How are we ensuring that we're giving the right dose and the right LNP to the right target? What exact transcription factors are we going to mix together in those LNPs? Once we believe we've optimized our prototype drug and we have a clinical drug candidate that we want to test in humans, we will then need to complete a number of enabling studies to allow us to move into a first-in-human study. These will include large animal trials, so looking at maybe some efficacy, but looking at pharmacology. So where does the drug go? How is it metabolized in a larger animal that's more relevant to humans? We will also have to do toxicology studies and safety studies. This is a very novel mechanism of action, and so we have to make sure that this drug will be safe to take into humans. We'll also have to prove that we can manufacture this drug at a scale that would be appropriate for a clinical trial. Once all of that is complete, we'll be ready to engage with regulators and seek to start our first clinical trial. We anticipate the first drug will be an mRNA payload that rejuvenates hepatocytes coming out of our metabolism program. Again, a likely sentinel indication is alcohol-related liver disease with the potential to expand more broadly across metabolic syndrome and chronic liver disease. At the same time that we're progressing our first prototype drug towards the clinic, we will continue to develop additional prototype medicines and validate them in preclinical models. So far, we've onboarded 2 cell types to our discovery engine. Jacob told you all about them already. The second was onboarded in less than 9 months. There are still hundreds of cell types left in the body that we want to pursue. Over the coming years, we will 2x the number of programs that we have in-house, and each of those cell type programs will generate therapeutic assets in the form of these prototype medicines. Over the next few years, we will generate 2 more of these prototype medicines. We will also continue to invest in supercharging our discovery engine with a goal of 10xing the number of TF sets that we've tested over the next few years. We've already generated the world's largest age reprogramming data set, but we're not going to slow down. We will achieve this 10x metric by continuing to bring down screening costs. As has been mentioned, we've brought down the cost of screening a transcription factor by 2x over the course of 2024. And we will continue to invest in leading the field in AI models. This ensures that we continue to grow our data corpus in a meaningful way and increases the value of every future experiment. Lastly, we want the hits to keep coming. We don't just want to screen more transcription factors because we think it's fun, although we do think it's fun. We want to generate more hits, and for that, we will need to maintain our lead with best-in-class AI models for cell age reprogramming. We've already seen that our AI models increase our rate of discovery, and the more data we add, the better performance gets. For each experiment we run, we can discover more hits if we test AI predictions than just our guesses. If we combine this with improvements in our technical approach, we expect to see significant gains. This increases our hit rate and ultimately increases the value of each dollar that we invest in our discovery engine. At the end of the day, we want more hits. The more hits we have, the more shots on goal we have, and the greater our chances of making multiple meaningful medicines that help people live longer and live better. With that, I will turn it back to Brian to close us out.
Well, thank you, Cathy. It's just so exciting to see what can happen when a smart, talented group of people come together and work really hard on an ambitious mission. In a moment here, I'm going to open it up to questions from the audience and from the live stream and invite some colleagues on stage. But I thought I'd just wrap it up here by talking a little bit about the company and NewLimit and who we all are. Everything you saw today that was presented, it was really made possible by the team. They're the ones doing the work. They're the ones putting in the long hours in the lab, debugging the code to make these things possible. And I hope you'll all join me in just giving them a big round of applause right now. And I also want to give a shout out to our early investors. Some of them are here in the audience, and it wouldn't be possible without people willing to make a bet on really ambitious ideas like this. And so, the venture capital industry has a big role to play in this of trying how do we advance civilization? We have to make big ambitious bets happen in the world. NewLimit is a bit of a unique company. We don't actually have anybody in the C-suite. One of our values is that everyone executes. And so everyone you see here on the slide, they're all doing a individual contributor aligned job with at least part of their time, even our managers. All of them are in the lab doing science, they're writing code, and it's a really special culture with a really ambitious, high potential group of people, an elite, I'd say it's an elite team that's come together to make this happen. And so it's really, really exciting. And I want to end here with a little bit of a call to arms. If any of you out there want to join an elite team working on an ambitious mission with the potential to change the world, I hope you will reach out to us. I do think this is one of the most important things really that can be worked on for Team Human. And we've got to make sure that we enhance the human experience and really try to figure out our biology over time. 56 million people die every year due to aging. It's arguably the largest source of human suffering on the planet. And I think we're at the cusp of being able to maybe make a dent in that. With that, I'm going to invite some colleagues on the stage, and if any of you in the audience have a question, we'd love to get to it, or on the live stream as well. Let's come on up. You can do 2 rows if you want. I'll try to get it all in frame. All right. Let's see. Does anybody have a first question? Anybody want to be brave and go first?
Don't be scared.
All right. We have a runner here with the microphone as well.
Are there universal transcription factors that help all cell types, or is each recipe completely different?
I really hope there are universal transcription factors. I don't think we have great evidence they exist today, but that's a potential upside case.
Yeah, I mean, it might be possible that maybe talk about the cell lineage and
Yeah, sure. So we think that it's quite plausible that cells that have common developmental lineages could share some transcription factors that have rejuvenating effects. So as an example, if a transcription factor rejuvenates a T cell, maybe it's more likely to rejuvenate a B cell as well. They share a lot of their chromatin architecture. Whether or not there are some that work on every cell type in the body, that one's a bit more out there, but I'd probably put my bet on the former existing.
Go ahead, yeah.
So in terms of the more hits, what are you thinking about feedback to when you'll say we won't get any more out of expanding more data sets? And does more hits mean a better medicine for the liver, or does it mean moving to a new cell type? And when would you make that switch?
I can go. So
Go for it.
it's all of the above. And so, even if we find initial hits in hepatocytes, we don't know if they're the best. And so we want to keep running screens there, continually improve, and then also do that in additional cell types. You asked a kind of maybe a leading question, or the question behind the question is well, are these hits actually any good? They might just be making the cells look younger, but are they actually predicting whether or not this is going to be a good medicine for people? Is this going to have dramatic functional effects on people? Today our models are more measuring that, predicting that look-like screen, that expression profile difference. But I think in the coming years, we'll be using more models to predict functional differences. And then this is going to be really valuable if we have a transfer learning between cell types. So as we move into some cell types where maybe screening in them, accessing primary human cells is very hard or near impossible, and running functional assays don't exist, it's hey, if we could actually in silico come up with small sets of transcription factors that could predictively affect the function of these almost unseen cell types. That's the ideal case of where we want these models to get. The models aren't going to be there next year, but in some coming years, that's what we want.
I've really had a question about the hits, which is that are they clustered in TF space, or are you finding them all over? Or is there a sign of programmed youthfulness, or is it that, there's something that you add a bunch of entropy and you get back to the state you want?
It's a great question. Is Nick here?
Yeah.
You want to give Nick a mic?
This is on. Yeah. Cool. They spread mostly over a good amount of the TF space, I would say. There are certain TF spaces that we have not found a hit in, but we found hits across a wide breadth of the TF space thus far.
One counterfactual hypothesis you might have is maybe the Yamanaka factors are super special and everything you find is very similar to a Yamanaka factor. SOX3, POOS6F1, KLF5 would not be the most exciting combo of TFs to find. It turns out that they're not clustered around the Yamanaka factors in any significant way. It really seems pretty broad.
Let's go here and then over there, yeah.
I'm really curious. It sounds like you guys have found some really interesting assays for functional improvement as well that you've noticed. Can you talk about how you'll think through whether these will have deleterious effects or off-target effects. Like, so they might have functional improvements in liver, but might not in another organ, which clearly we need to do both if you wanted to put this in the body. And I know you're going to do toxicology studies eventually, but just I'm wondering upstream of that, if there's ways to kind of incorporate that.
I think the first point is just in how targeted we're being, right? So the expectation with these mRNA medicines in LNPs targeted to the liver is that you're only going to be turning on these transcription factors in the liver. So I think we already have a head start compared to a small molecule in terms of off-target effects. We will have to do all of those toxicity studies before we can get to the clinic to show that it's all getting taken up into the liver, and we all know no delivery method is perfect. So even though the bulk of it will get into the liver, there will be some in systemic. Is there anything you want to add, Rowena?
No, I think that.
I think one interesting thing that you can find as well is most TFs just like don't even have an effect. And that's partially because like the chromatid's just not open for them to access those cell types. And so we think that by kind of stacking cell type specificity and everything from the TFs that we select to the chemistry of the LNPs to how we engineer the mRNAs can stack further and further where we get more and more cell type specificity out of it. It still is not perfect, but we think can get you a lot of the way there of like reducing those off-target effects.
And to be clear, we haven't done all that work yet. So you've correctly identified an important next step. I think we had one here, Matt, and then, yeah, go ahead. Can we get a runner over to Matt?
Sorry.
Okay, if you're ready, go ahead. Yeah, sorry.
Yeah, I was wondering if you have explored how long the reprogramming lasts, especially with the LNP system. So once you have your hits and you achieve the reprogramming and, let's say, rejuvenation of the cell, is this initiating a cascade of events in the chromatin dynamics and sustain the young state, or you need to keep treatment, I don't know, for all your years because you are just introducing RNA, and if not, the cell will go back to its original age?
Yeah, it's a really astute question. I think, obviously, a medicine you only have to take every once in a while is way better than one that you have to take every 10 minutes. And so these effects are much more interesting if they're durable. So to answer your question, we've done a number of experiments to try and measure how durable these are, but maybe not as many as we hope to in the future or over the same timescales we hope to in the future. So in some of our longest term results, we found, for example, that in vivo in human hepatocytes, some of these effects can last several weeks, similar to the duration between doses in current commercial LNP RNA medicine. So we think we're in a range where the effects are durable enough that one can imagine making a medicine even if they didn't get better. But that's certainly not the limit of our ambition. We would want these to extend out, and ideally you'd be in a place where you could have medicines that were activated once and lasted months to potentially years. We know in the limit case that is possible. We can make targeted epigenetic edits in specific loci, and other groups have shown in non-human primates, for example, these epigenetic edits can last years. Or in cell culture dishes, you can passage cells 400 times and they still preserve the edit. So I think from first principles, that sort of durability is possible on much longer timescales.
Matt
Okay.
and then Mark.
Let's get another one here.
Yeah, really impressive progress for all of you guys. Congratulations. It's a kind of similar question for me. I was curious if you think the mRNA expression duration is going to have to be something to optimize with what you're working with, or just by the nature of partial reprogramming, it'll probably be enough of a pulse, so to speak.
Sure, I can start. So yes and yes. We think part of the beauty of epigenetic reprogramming is that you don't have to like durably express these transcription factors. In fact, it's a feature that they're only there for a pulse of time, a few days, and then they're gone. And then the effects, to Jacob's answer earlier, are durable, and there's a question of how durable yet. We just haven't run the long-term experiments yet, so we don't know.
Okay. Go ahead.
Very impressive. Also, some questions about the biological mechanisms. First, and I think they're all interrelated. How many transcription factors in your hits, 2, 3, 4? Do you have any evidence that they act sequentially, or do they act simultaneously? And do they act on the same genes? You have a bunch of them acting simultaneously in a synergistic way, or is it that you have to they each activate different genes that then work collaboratively?
I'm happy to address these quickly and would love for any of my colleagues to jump in too. So on the first point, how many TFs are in the hits? We do find examples of hits that have even a single transcription factor, but the majority seem to be combinations. There seems to be a number of more frequent interactions between transcription factors than you would expect for normal gene-gene interaction graphs, which tend to be pretty sparse. And so often our most exciting hits actually have combinatorial interactions between those TFs. And then they also have more effects together than they would alone, even just additively. So the majority of our hits are combinations of one flavor or another. I think your second question was around, do they seem to affect the same biology, or do they seem to actually hit the same sets of genes, just stronger? We don't have this incredibly rigorously estimated, but I think it's much more the former. You see broader gene expression programs involving more specific targets moving when you have combinations than you do when they're alone.
All right, go ahead there in the back, and then Mark, I'm not sure if you had one too, but go ahead.
Sure. Can you overdose on these transcription factors? And by that, I mean, can you give so much that you actually make the cell too young and lose what you're looking for?
Wes, do you want to take that?
I think the answer is we don't quite know. Certainly, you could give too much of a transcription factor and change the cell more so than you'd hope. But I'm not sure we know the answer quite yet.
Yeah, I mean, maybe you want to talk about the therapeutic window, say versus the Yamanaka factor or something.
Yeah, I mean, part of the motivation for coming up with novel factor sets is that even if, let's say, you accidentally dosed 2x or 3x or 4x your target dose, we would want the cell reprogramming to be resilient, where you're not affecting the identity or like shifting to some toxic state of the cell. And so that's kind of built into our screens to a degree. There's almost like a third axis on that screen of like cell identity, age, and then like we're building on the third axis, and we don't have it now, so we weren't doing 3D plots yet of almost like a safety axis. And so that's all kind of in the works.
I'm so excited for the 3D scatter plot.
Yeah. All right, who's got a microphone? I see lots of hands, but who's?
Do we want to move online, or do we want to?
If you have you got an online one you want to shout out? All right, let's
take an online one.
Let's go to the live stream.
Do you anticipate running into difficulties with targeted LNP delivery as the company expands with cell and tissue types?
I think the idea, the reason why we've really focused on the liver is that there's an LNP that's targeting there. It's clinically proven, and so it's just nice to have that sort of stability proof of concept. I think we're hoping that the technology really is going to catch up to the other cell types, and we'll be able to deliver in a safe, effective manner.
I think I hear Blake say all the time, we do not intend to be an LNP company. That is not our goal. We intend to work with partners and leverage the technology that is available and that is advancing. We are very intentional about how we pick our cell programs, and is there a commercially viable medicine? Is there a small sentinel indication? Can we actually do this from a technical standpoint? Is there targeting technology available? And maybe in the future, we'll have to do some of that ourselves, but at the moment it's, trying to leverage what's already being developed.
I have another one here. How long do you think until the average person would benefit from these medicines?
The average person. The average carrying a lot of weight here.
Non-diseased? Yeah.
Cathy, what do you think?
I mean, clinical trials take a very long time. So anyone who's sitting in the room that works in drug development knows it is a long path into the clinic, and it is a long path out of the clinic. I think the beauty of our approach is that we hope to get these medicines to patients with diseases, which you can almost think of as an accelerated aging, within the next decade. Getting to the point where the average person is taking this as a longevity drug, it's going to take a lot longer, but we have to start building the puzzle pieces of the individual indications, and then we package them all together and you get a wellness IV that's your longevity drug.
All right, got one here in the back.
As you train the model on more and more internal data sets, are you seeing that the learning rate of the model is slowing down? You've saturated what you can learn from these in vitro systems. And if so, are there ways you're adapting the way you're screening and generating the data so you can maximize the bits of new information per dollar spent?
Yeah, that's an amazing question, actually. Yeah, we do see for a given model that like the model learning starts like going down as we like put more data in the model. But we don't think that this is like an issue. This is actually more of a feature because you can also adapt the model you're using to the data set that you're using. So if you think about all the like modern machine learning that is happening right now in text, a lot of these models, if you were to train them on a small data set, they would perform actually quite poorly. But because they have the expressivity and they also are trained on very large data corpus, they really shine. And we believe that the same will be true for our data corpus, where at a certain size, we will have to like maybe go to the next generation of models so that we can keep having the performance go up.
Okay, yeah. Let's get one here.
What are some of the key limiting factors for you to push the scaling law, as you mentioned? If you stack rank them, what would be the top three?
Is that you again, Lucas?
I think this is roughly experimental, so it's largely around how quickly can we generate data. I'd say the number one is the cost of generating that data, and so at least as of right now, we think we're better at picking which questions to ask than anybody else because our models are already pretty good, mostly thanks to Lucas. But nonetheless, there are still real costs there, and so something Barak on our team has really been working on is driving those costs down over time. Following the cost of actually just doing those readouts, I think the main cost on the front end is that right now the system we've built is really focused on mimicking an LNP RNA drug, which might sound simple to say, but it turns out it's actually quite complicated to build in base pairs. And that means that on the front end, we also spend a bit of time having to create a lot of the reagents for each of our experiments. So we're working on some technologies now that James is leading actually to be able to do a lot of that work in pools as well. So say those are the number one and number two main blockers to simply going from 10x over the coming years to 100x to 1,000.
Yeah, if you think about how we could 10x the size of our data set, yeah, some of this is about novel chemistry that the team has come up with 4 or 5 different innovations that have stacked on top of each other that have allowed us to run these screens at larger scale or lower cost. There's also, you saw maybe in the welcome video, we've started to introduce a little bit of automation with robots and liquid handlers. Part of it is also a company culture. I think we might be one of the only biotechs that we use an OKR system to really, and with clear owners every quarter, to drive down, let's say, the cost of this one step in the screens by 25%. And we've tried to be very rigorous about optimizing each step in this process. And so, I mean, who knows? Someday we might go build the gigafactory of transcription factor testing or something and have an army of robots. But we've only scratched the surface of the search space and the number of cell types. So I think we could really scale many orders of magnitude from here.
I need to correct Brian's answer. The Gigafactory is coming. It's not a maybe.
All right. We got one here. Yeah, and then let's get one in the front. Go ahead.
A tissue I deeply care about is the thymus. It has a pretty meaningful role in the immune system, and also as we age, it goes through evolution. And very curious if you've thought about going that direction in thymic regeneration down the road or in the near term with these technologies.
Clearly no one has.
I'm happy to take it. Yeah, it's definitely something we've thought about. It's, for various reasons, not one of the top 4 cell types we're going after, but that's not to say because we think it's a poor idea, merely that there are various logistical and technical choices that go into some of the cell types we choose, like do LNPs exist that can actually take payloads there? Do we think we can do screens in that cell type? How big is the patient population? Or are they at a stage of their disease where they'd be willing to take a medicine? I think the thymus, unfortunately, scores kind of poorly across some of those criterion, but it doesn't mean we wouldn't want to expand there one day. One reason we are really excited about T-cell rejuvenation is because, at least in humans, unlike in a lot of model systems, your T-cells really are like a stem cell pool. And while your repertoire does decline with age, it doesn't seem like you have many holes emerging in your repertoire. And so we think if we could fix even the T-cells you have around, you'd get a really big benefit. Now, if you could get even more new thymocytes, it probably is additive as well. So maybe that's like the V2 of the immunology program.
Okay, great. I think we have time for one more question, and then we'll move to our reception upstairs, and maybe if any of you want to see the lab as well. Go ahead, please.
I have a question about the direction of reprogramming for similar cell types. So a bit along the lines of the T-cell, B-cell discussion from earlier. You mentioned repurposing the same formulation of transcription factors for different indications. So for example, alcohol disease versus a genetic liver disorder. How do you all think about how hits for one indication in a single cell type translate to other indications in the same cell type? And does the modeling inform that, or is it more screening more donors?
So my initial answer is relevant to hepatocytes. So coming from GSK, I led a large clinical development program on a genetic target called HSD17B13. And it's a naturally occurring mutation that protects patients from developing multiple forms of liver disease. And I think the nice thing about it is it gives us a genetic proof point for a perturbation only in one cell type having an impact across the organ, and the same perturbation having an impact across multiple diseases. So you could almost imagine that our hepatocyte reprogramming drug could become an etiology-agnostic cirrhosis treatment.
So in layman's terms, we're trying to make the cells act younger, which could help them be resilient to all kinds of disease. Now, whether we can accomplish that or not is we still need to do further study, but I think we're well on our way. So with that, let's end it here, and we're hosting a reception upstairs. We hope you'll all join us and can answer more questions live and get a little tour of the lab. So thank you all for coming and supporting this exciting research.
Thanks so much.